Cell signaling pathways are how our cells talk to each other, and it allows for appropriate responses from our cells. There are three main components of cell signaling pathways, and they are reception, transduction, and response. Reception can be thought about as a keyboard. The signaling cell produces a signaling molecule, and that signaling molecule is recognized by a target cell by a receptor protein, and it attaches to the receptor protein. Once the signaling molecule attaches to the receptor protein, or once we type what we want to do on the keyboard, the change in the receptor that triggers a series of changes along the pathway is called transduction, and we're going to think about this as the computer. We can think of response as a printer. Transducted signal triggers a specific cellular activity. So the activation of cellular responses finally occurs. Just like the printer prints out the paper, when you type it on the keyboard, you type your document, the computer finalizes it and it prints.
Okay, let's talk about the deep protein cup receptor. We have our cell membrane here, and the cell membrane we have the extracellular and the intracellular. This is our DPCR. So we know that the most characteristics of DPR is because they have 7 transmembrane alpha helix. And this is our G protein. G protein is this special protein that have ability to bind DTP and GDP, the guanine diphosphates and guanine triphosphates. So all have a subunit alpha, beta, and gamma. So in this structure like this one, they are in inactive place. When they are in inactive moments, alpha subunit is bound to DDP. Now, when the ligand bonds come to the DCPR undergoes to give a conformational exchange. This conventional exchange is characterized by this bounding under the DCPR. This bounding is the conformational exchange. Now, the alpha subunits will dissociate from beta and gamma. When they are dissociated from beta and gamma, it's gonna change. Alpha helix will change the DTP to DTP. We change GDP to DTP. And then when they are dissociated, this is beta, this is gamma. They are separate from alpha. So these both will try to find the another protein to be equal. They can do it, so alpha will find this protein. This protein is called the target protein. This is the system. Now, this is not gonna stop like this. The system will try to be normal. When it tries to be normal, that means that the target protein, that one here, is gonna give a new signal via the second messenger. And finally, the DTP hydrolyze to give DTP. When they hydrolyze to give DTP, it means that the GTP it means that we're gonna have like this A GTP will give more GTP plus phosphates. So it's gonna be hydrolyzed. So after this one, after the G protein coupled receptor, we're gonna talk about the G protein linked receptor. For that one, this is our cell membrane also here, and this one is our G protein linked receptor. This is our ligand bond. When the ligand bond come to attach to our G protein linked receptor, this immediately, and this is our G protein, and when the G protein is inactive, is combined by the DTP. So when they are like this one will go, they're gonna give the conformational exchange. When they give the conformational exchange, the DDP will move to this, to the G protein link receptor. When they move to this one, it's gonna change it. At first we change GDP to GTP. When it change to GTP, it's gonna now diffuse in the cell membrane to fix on the enzyme. When this is our enzyme, and in many process it's gonna give our response via this one, via the cyclic EMP. And after all, it's gonna be in the same place. GTP will be hydrolyzed to give